Alright, in this video we are going to learn about how to find a gene. Now this isn't going to work in all species. Certainly not in something like a bacteria. They don't use CpG islands. And if you don't know what a CpG island is, you need to go back and watch the video on development and differentiation and how histones and DNA interact, and how you methylate C's followed by G's. Now, in humans, it works pretty well. I'm going to show you how to find your likely location of your gene. And now in EHUX, it's not going to work as well, but that EHUX is the name of the algae we're using for our project 3. It's not going to work as well as it would in humans, all right? But it will work. And I'm going to show you here, and I'm actually going to help you out. I'm going to go through each of your genes and we're going to do this same exercise for all 6, and you'll get to see what's going on in your particular sequence, and I'll give you some hints. Now, what's going on here in this sequence? Well, this is the one we recognize as the ref-seq on chromosome 3, that 8,000 base pair that has 2 genes, one of them being GPX1. So this is a human. So how would we go about finding the CpG island in a human? Well, you would think easy enough. It's a C followed by a G. And if you look at this, this works reasonably well in human DNA. We know there's a gene up here, but we don't know exactly where it is, but we might not have the complete sequence, the promoter sequence before that gene. But as we look through here, well, it's kind of randomly dispersed, as you can see. But then we get here. This is a CpG island, all right? You do not get that many C's followed by G's without being in a CpG island, all right? And I scroll down, there are a few more down here. But here is the beginning of your gene somewhere in this region. Now, a CpG island can extend all the way into the first exon of a gene. So if you only have one exon, it can encompass the whole gene. But generally, it only encompasses through the first exon. And it will, the older the gene, the less C's and G's there will be in the exon part, in the coding part. So a very old gene won't maybe have any C's and G's at all. A young gene that has only recently evolved may have C's and G's in the actual exon. But generally you find it in the promoter, pre-promoter, and even extended promoter, which we will define here eventually. So here is our probable promoter for the gene, and somewhere in here, and you've already looked at this one, somewhere in here is our start codon, all right, and our transcription start site and things like that, and our whole gene should be in here somewhere after this CpG island. So here's a good way to find your gene, all right? It's going to come near this, the beginning of your gene will start near this particular sequence. In the middle of it, hard to say, because the first exon could be in here. This could be the first exon right here. You really don't know, but you know that there is probably a gene in this area. In fact, it's almost 100%. Now here's our problem. I did this with C followed by G, but we learned that this is an RNA. So I should not be searching for C's and G's, should I? I should be searching for G's and C's. Now it works pretty well because, all right, you still can detect the sequence here, all right, with G's and C's, all right. So there are way more in this region than there are in the ones, but it's not as good. And one reason for this may be that we're going in the opposite direction, the direction of this sequence, but no, it's forward. So, but here is your CpG island, definitely in this region. See how many G's and C's. Now, I found that it's better to search in humans. This may not work in other species, but in humans, to double it. You rarely find two CGs next to each other. So in this sequence, you would rarely find two GCs next to each other. So what might be better case to do is look for a GC-GC. And there you go. We narrow it down much better. Very easy to see where our CPG island is. Our CPG island is right here. All right, you can see there's one lone one down here. There are three up here, maybe a kind of a straggler up here, but definitely a lot here. This is your CPG island. So I'm going to add a third one, and we'll see if that's useful at all. And yes, it only shows up one place, right here in the middle of our CPG island. As you can see, there are two if I take off one, and then the others appear. This is definitely our CPG island. Now remember, this is RNA, so normally, of course, you would be using CG, CG, CG. And if you do that, it doesn't show up at all. All right? So, which is interesting. It was rarer for the opposite, but the whole idea here is CPG islands are what you're looking for, and I'm going to go through this with you to help you find your gene in your sequence as well. Now, it's not going to work as well with EHUCs as it does with humans. It's a very easy trick to use for finding genes in humans and in mammals in general. Doesn't work at all in insects because they use a completely different sequence than CG. But plants generally do use CGs, and actually a couple of others. And in algae, they use CGs. So it's still possible to use it to find your gene. So I'm going to open up. Let's see, where is this? Right here. And I'm going to start with EHUC's sequence 1. So whoever has this one, you need to pay attention. And so first thing we need to notice is that there are some letters in here that we're not used to. This is raw genomic sequence, and they intentionally leave it, and it's not sequenced well. They didn't have, a $500 million to sequence the EHUC's genome. They had, maybe a couple of million. So it is not sequenced well. So they give you as much information as they have. And what they did was, whenever it's like questionable what the sequence is, they leave it small letters. So you can look at it as when it's a capital letter, they're fairly certain that that's the correct sequence. When it's a small letter, maybe. So what are we looking at? We also have ends. Ends mean the sequencing didn't work at all. All right? And you can see around that they're not real confident of what's around it either. All right? Now, this means that you will often get a sequence like this, and it's a mess. And there's nothing you can do about that. So we have to learn to work around that and be able to accept some fuzziness in our answers, which is easier for you because it means as long as you make a good argument, I'll accept it, and then you can go and move into the wet lab and find out what's really going on. So what do? Well, we go and, of course, let's search for C's and G's. Oh, man. This. Now, we're assuming this is genomic, and it is in the DNA, so you would search for C's and G's. This isn't good, and this repetitive sequence is probably wrong. They probably should have put ends. There's no way that this is something that's legitimate, probably. So you can see that this is bad sequence. They know this is bad sequence, and they put that in, so there are ends there. So with this being bad genomic sequence, this is not a good measure, just C followed by G. So let's try 2 CGs. Does that help? A little. All right, we see that there are regions in here that are better than others as far as coding sequence. As far as CPG islands, we can see that, okay, this is getting better. I am very sketchy about these places where there are small letters, all right, especially this piece right here. So let's try another one. Let's try 3 CGs, and now we've narrowed it down somewhat. Almost none down here at the end. We get a few here. We get a few here. So I think your best bet, those of you who have sequence 1, is to look for a coding region, all right? Some that might start somewhere in here. This is low quality here, but you might start looking for a gene in this region, or you might start looking for one in this region. I suspect maybe there are 2 genes here. Maybe there's a small one here, and there's a larger one here. So for you, I would probably, if it were me, I would start with this region. It just looks better. Even though there's this big, large chunk of ends here, I would look here for my gene. So you would think then that this is your promoter region, somewhere in here, and this is probably your gene all through here, maybe down into we don't know where it would end, but you can see that there's some bad sequence in here. Not just small letters, but actual ends where they know there's a nucleotide, but the equipment just couldn't decide what it was. And this often happens when there are repeats, and you can see that there are repeats. You should be very, very cautious about repeats. Any place that there's repeats, be very cautious. And you probably can't find a gene there anyhow. Even the computers aren't going to find genes there. So, as another hint, knowing that how the sequencers work, you want to look for regions. You're not going to find one. You're only going to find it where there's probably pretty good sequence. And so you might start, if this is a CpG island, I would guess that there's a gene right in here, mainly because there might be other genes in here that you can't find because it's bad sequencing. But I would say anywhere in here is probably a good chance that you're going to find a gene somewhere because it's near a high CG content. So you've got 3 or 4 of these here. This is probably your promoter. So if you're looking for a promoter region, this is probably your promoter region, and then there's a gene after it down here. So that's how I would start out if you are in sequence 1. It's not perfect, but that's what I'm trying to show you, is that this is not an easy thing to do. The computer went through, and I'll tell you how I found these. I used the computer-based system that went and found these sequences and predicted that there were genes in here. And someone has gone through for a few of them, all right, and it's not published, so you're not likely to find it, found that there is an mRNA that matches to some of these pieces that they found in EHUX. So that means there is a gene, at least one gene on this length. Now where it is located, it could be down here, it could be up here, I don't know, but I would look for your 2 genes. Either one would be in this region or one would be in this region. With where the yellow CG is, that would be where you would look for your promoter. All right, possible promoter here, so possible gene here. Possible promoter here, so possible gene here. That would be your best guess. You need to go figure out which one you want to work with. This is actually a really good sequence in this area, but then you run into really rotten sequence here. There's no way that this is legit. And then there's region here, and then you get some good sequence here, but you run into some ends. So I would look at those 2 regions if you have this sequence. All right, so now let's move on and see what we see for, let's see what we find for sequence 2. Let's see. Where's sequence 2? EHUX gene 1, EX gene. Or was that 2? Okay, this one is 2. The one we were just using was 1. Yes. All right, so let's do the same thing here for sequence 2. It looks a little cleaner. Sequence 1 is difficult because it's not very clean. There are some ends and lots of little letters. This one seems to have a few more larger letters, and so that means they're more confident about the sequence being correct, and we don't see any ends at all, so that's good. So let's start out with just CG and see what we can tell. Well, we get lots here. It's hard to tell with just CG, so let's do CG. Well, still not great, though we see a region here that doesn't have many CGs, so that actually is telling you that this is a more likely place for a gene, but there's some bad sequence in there, so don't know. So let's add another one. And here we narrow it down a little more. So we get some CGs here at the end, but that would put our gene outside of the sequence, and I'm telling you there's a gene in here. So if we have some sequence here, I would say this is your likely promoter. All right, and your gene is probably going to be somewhere in here. And it's a good empty space between your two CGs. So there's probably a promoter here with a gene starting after this. And this is your gene. So you would look in this region for your gene. All right, now I don't know where it's going to be. It's up to you to figure out where a potential reading frame might be. And this is not going to be easy. So you'll have to decide amongst yourselves which region you want to use. In this one, it's pretty clear that you want to use this region. Now realize your CpG island is, if you know that this is the region you want, the CpG island can go into the first exon. So let's reduce this a little bit and just kind of do it that way. Yeah, that's looking... So when we do this, it's here, all right, right here between these. And if I reduce that, you see, yeah, that's probably your CpG island. This is probably your first exon, all right, if you can see in here. And your gene is here. You get some bad sequence here, so that's going to throw a monkey wrench in there. But that would be what I would do first. Identify a good region, and I would say that's probably because you have a lot of condensed C's and G's in here. This is very likely your CpG island for sequence 2. So those of you with sequence 2, that's where I would begin. All right, now let's go on to the next sequence, which is sequence 3. And we'll do the same. Now this one is a bit longer, and we've got some ends. We've got some bad regions, but it looks like we mostly got some good sequence in this one. So let's go CG, 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 and let's see where we end up. Well, what looked like a lot up here ended up only being 2. We got 2 here. I'm thinking this is your promoter, probably this region here. Maybe we're moving into the first exon. Maybe it could be a long exon. Or maybe we've moved on to another gene down here. So somewhere in here, I would say this is definitely a promoter. So it might be a small gene somewhere in here. And this looks like good sequence. This isn't as clear a signal as good sequence. Let's try something. I'm going to put another CG in there. And they all disappear. All right, so not a common thing. Here are your others. Let's go and take another 2 out and see how it looks. Yeah, we get lots of concentrated CG in this area and less so up here, but this could all be promoter. This could be a very large promoter. And then down here we have our gene, and it could, the problem is maybe we have a single exon, no introns at all. And this is just one. This looks like it's probably the beginning of a promoter for another gene. So I would be looking into that area where I found the most of these. And this in particular is suggestive. If this is small letters, but if this is correct, then I would definitely say this is something to look at. And you have 2 pretty close CGs here. I'm thinking that this is the region you should look at as your promoter, and the part down here as your gene. Okay, this might be part of a first exon, and then these you can't really trust. All right, so that's the way I would look at this sequence. All right. You get to see how just difficult this can be. The computers just guess. And it's hard enough. There's no way. They just guess. So now we go on to 4. And ooh, this is just looking ugly. This beginning part is just ugly. And we get some ends, but we get some good sequence in between the ends. So let's see what we see here. Well, I've got 3 CGs, and I don't get anything. So this one is very low in CG content. So here we go. Lots of CGs here, a few here. I would think this is your promoter. This is a potential promoter down here maybe. This probably is not enough, but you never know. I see CGs all around. But this is your probable promoter, and this is your gene. But you shouldn't get too many CGs inside a gene. So let's reduce it again. Now, if I just go CGC, this is just crazy. That's why don't believe any of this. The sequencers aren't very good with repeats. And to me, it looks like you get more CGs in this area. And this is the promoter. Then you get ends, and the ends just throw a monkey wrench in it. But it looks like probably the start of your gene is near these ends. But we get some good sequence right before it, so that's a good thing. And then we get good sequence after it. So whatever is going on here, maybe it was a glitch, and hopefully your gene is just in here. But then you get, and this would be maybe the start of another gene. There's no telling, but that would be my best guess. All right, for you to start. This one might be actually straightforward in that, well, here's my CpG island, and here's my gene, and then here's another CpG island. So you'd look in here. That's the best advice I can give you. You can go, this is nothing. Ignore this, ignore the ends. You're going to have to look at something else to find your gene around here. The ends don't mean anything, but you do see a big cluster here. I like this. Even though these are not for sure, this looks like your best bet for a CpG island. Yeah, sure enough. And then you would look maybe for your gene here where there are fewer. There is no other way to work with this sequence. Bad sequencing, but try it. All right, now we move on to sequence 5. And this one looks better. Kind of long. So let's go with just CG. What do we see? It's just kind of messed around all over the place. We have this. We have lots of these. Let's see what we can find here. 2 CGs. This looks interesting, but they're small letters. I see periodic CGs. All right, a little better, but still not enough to say for sure. But when we get to just these, it looks like pretty clear that it's either here, even though those are not good. But this looks a little better. These are clear-cut CG that are sequenced well, they're saying. So I'm thinking this region, if we agree with those are, then this region is your CpG island, and maybe your gene, maybe your first exon is down in here. And then this looks like maybe there are no CGs. This might be the later part of your gene. I would definitely go with that first. Doesn't mean it couldn't be somewhere else, but notice even in the good sequence, I don't get any CGs up here. None at all. Look at all the good sequence here. No CGs. All right, so I think that might be coding. It might be another gene, something like that, or it's just a region that doesn't do anything. I would say this is your promoter, and this is probably your first exon, but there is no way to tell. You got to go and look at it some more. So this would be where I would start. All right, now let's go on to the last one and see what we find. All right, so we go in here and we look at this, and I am not, we get a few, let's see, I'm doing 3 CGs, and you can definitely tell where your CpG island is here. I think this may be the easiest one. Here's your CpG island, and maybe there's another one for another gene that is after this. But I would say this is definitely CpG island. Let's reduce it a little and see what it looks like. Yeah, definitely CpG island, maybe first exon, and maybe that's the end of the gene. You've got pretty good sequence here. I don't see any ends. Yeah, this one might be. This one is probably the one that I would be the most confident on, even though you get. So let's see if we add a couple more CGs. So we know what that looks like. Let's add another one. They completely disappear when you get that many CGs. Yeah, this looks too good to not be a CPG island. This looks kind of like first Exxon. So that's the way I would look at this. Now realize CGs can be in the introns. They can be in your other exons. Just you shouldn't see them as often as you see them in the first exon. So maybe this is the promoter, and this is your first exon. It's hard to tell, but this region is where I would definitely look for my promoter to be. All right. Now we will find other things to help you clue in on where your gene is, but as a group you should watch this video and then kind of say, well, where do you think it is? And I've given you the hints essentially where I would start. So I would start with the one that I have hinted at, and then you would you move forward. And the next thing to do is to take the region that you think is coding. So we'll talk about that, but you'll take the region that you think is coding and you'll copy that and do a blast, and hopefully you'll find it match some other species for some piece that is highly conserved across many species. Something that is sequenced well, something maybe an algae that is they've basically done better than this one. All right, so the whole idea is this was one that they didn't have a lot of money to work with, and often you will run into that. So if you think there's a gene in here somewhere, our next part to help find where the coding region is to take our piece that we think where the gene is. So maybe I start in the middle of my CPG island and go down here to maybe this. Take out this piece and we blast it against an algae that is well sequenced, or a couple of algae that are well sequenced, and we see if it matches for any genes. If it has any sequence homology or similarity, we can work with it, and if you find that, then you can move forward. So that'll be our next thing to do. But the first thing you have to do is come up with a piece of sequence that your group can agree upon, that you want to start with. So that will be your first assignment, is to find a likely piece that you want to look at. And I've kind of given you, probably here on group 6 here, you guys, I've given you a big hint. That's essentially where I would start. But I think I've given all of you a pretty good hint on where you should pick out a piece in here to study and see if you can find some similarity to some other gene in a well-sequenced organism. So this is our first part in project 3. Get started on this, and I will I'll do a blast video. So once you pick out your sequence and you tell me that you've got it, you watch essentially the video I make on using it with blast, and that will be your next step to finding your sequence. I'll show you how to do that. All right.